Okay, let's start lesson 7, lipids and proteins. In this lecture, we are going to learn chemical structures and functions of fats, phospholipids, and steroids, saturated and unsaturated fatty acids, lipids as structural, functional, and energy storage molecules. Then, in the second part, it will be the protein chemistry, three-dimensional shape of proteins, allowing them to function as a structural and functional molecules, and at the end, enzymes as organic catalyst. Lipids include fats and oils with only carbon, hydrogen, and oxygen. Now, the ratio of this carbon and hydrogen close to 1:2 with less oxygen than carbohydrates. Earlier in lesson 6, you learned that the ratio is 1:1, but here the oxygen ratio will be less than that. And lipids are more hydrophobic than carbohydrates. Carbohydrates are hydrophobic unless they have the functional group modifying that part. Otherwise, they become hydrophobic. But this one, lipid, is more hydrophobic and they are soluble with lipid. So fat will be soluble with fat, not with, it does not go with water. There are 4 types of lipids we are going to discuss now: fatty acid, triglycerides, phospholipid, and steroid. All these lipids have diverse functions in our human body, which is very essential for us to survive. Let's start with fatty acid. It is the monomer of lipid. Monomer means single unit. Like that carbohydrate, we know the monomers like glucose, fructose, those are the monomers. Here, the lipids monomer will be fatty acid. And this is how it looks like. You see carbon at the center, then carboxylic acid functional group here, COOH. That is the carboxylic acid, COOH. And then the R group is here, the hydrocarbon group. These hydrocarbon group, it can be a chain, it may be long and include double bonds. So in all fatty acids, one thing for sure we are going to find is that COOH, that is carboxylic acid group. There can be 3 types of fatty acids: saturated, monounsaturated, and polyunsaturated. Saturated fatty acids, they are saturated because they don't have any double bonds, right? And most are animal fats. So if we find no double bond, that would be saturated. Each carbon attached to the maximum number of hydrogen atoms. For example, palmitic acid is a saturated fatty acid. Okay, so let's take a look at this structure of palmitic acid. You see, start with the carboxylic acid group on your left. You know this is the functional group, COOH, carboxyl acid group. Rest of the thing is a very long chain, which is the hydrocarbon chain, and all of them are carbon, hydrogen, carbon, hydrogen. And between the carbon, we see these are the only thing we can find is the single bond. No double bond here. Again, if these are all single bond, that is saturated. And most animal fats are like this. Not good for health. Unhealthy thing, fat, is this saturated one. If we find one double bond between carbon atoms, that will be as mono unsaturated fatty acid. Again, if there is any double bond, this is saturated. If it is one double bond, mono unsaturated. Mono means one. And most of them are found in plant oils. The plant oils are good most often because they are mono unsaturated. Example is here the oleic acid, C18H34O2. That is a mono unsaturated fat, and it is found in olive oil, which is the plant origin. And how does it look like? There you go. You see, the left side COOH carboxyl group is similar to all other fatty acids. And then when we look at the hydrocarbons, we see this double bond here. This is why it is unsaturated. And as there is only one double bond, that will be mono unsaturated fatty acid. Now compare that one with this picture. This is linoleic acid, but right now you see not only one, it is two double bonds you see there, more than one. So when we have two or more double bonds between carbon atoms, that will be polyunsaturated fatty acids found in seeds. That is also a plant origin. And these are good for health. I mean, if we consider which type of oil better for our health, definitely it is the unsaturated one. Mono or polyunsaturated are better than that saturated one we get from animal sources. Number one is done, fatty acid. Second thing is the triglyceride. Tri means three. So we will have three fatty acids here. Okay. So three fatty acids to glycerol, they will be linked together to form this triglyceride, a neutral fat. So this is how they look like. You see on your left side, that is the glycerol molecule. On the right side, three fatty acids are there. So when they combine, they make this reaction to happen. This is the anabolic reaction, and this is dehydration synthesis. From every one of those fatty acids, one molecule of water is removed, and the end product will be triglyceride. So three fatty acids, one glyceride, we call it triglycerides. It's a dehydration synthesis. And these type of fat, they are stored in our fat cells for insulation, temperature regulation, and energy source. So this is a good fat, you see, good for our health. Maintains that insulation, regulate temperature, and when there is energy needed, this fat will be burned to make a lot of those energies. Regarding the good and bad fatty acids that I discussed earlier, just to mention one thing, if it is essential polyunsaturated fatty acids, they like omega-3 fat found in flaxseed oil, fish, and important for cardiovascular health. So these are the good fat. Saturated one or trans fat, that is hydrogenated oil. These are bad fatty acids, which increase the risk of risk of heart disease. So these are bad fatty acids. So after fatty acid and triglyceride, third type of lipid will be phospholipid, which is the major component of cell membrane. And this is the classical representation of this phospholipid. So what do we see here? Starting with the right side of the slide, you see a large head. This is the hydrophilic phosphate head. phosphate, PO4, phosphorus, and oxygen, that's the phosphate. And these are hydrophilic, that means they are water loving. And those two tails you see there, these are hydrophobic fatty acid tail. So the head, phosphate, which is hydrophilic, tails, fatty acid, hydrophobic. So why is that significant? These structures, these phospholipids, they are composed of glycerol, fatty acid, phosphate with a nitrogen containing group. And at the end, when we soon talk about the cell membrane in a cell, you will find that these phospholipids, they actually are responsible for making this sandwich pattern, this um, cell membrane by phosphate bilayer that is formed by these phospholipids. At one side, they are polar and hydrophilic, which is actually facing the water side, the extracellular side, and inside the cytosol side. So in both sides, there are water. So this part, the phosphate part, will be facing that area. But hydrophobic, the fatty acid part, will remain hidden inside, non-polar and hydrophobic. So just to remember that the head part will be facing outward and inward, but at the middle, because this is a phosphate uh, bilayer, phospholipid bilayer, this hydrophobic part will be the inner side of the cell membrane. So we use this word as amphipathic. Amphi means both, because it has both polar and non-polar, hydrophilic and hydrophobic part. So these phospholipids are called amphipathic. Fourth and last type of lipid is steroid. And all of these steroids with four rings, and they will have be derived from cholesterol. Have a look at their face. Right? So four rings you see there. You can also see the steroid nucleus found in all steroids. And they are non-polar. That makes sense, right? Because lipids or fat are usually hydrophobic and non-polar. But these steroids are important, especially when they make steroid hormones. They are used for cell communication through the blood. Examples: testosterone, estrogen, aldosterone, cortisol. Look at those names. Very, very important hormones for our human body to function properly. Testosterone, estrogen, aldosterone, cortisol. Okay, so that was the brief discussion about lipid. Next part here is coming about protein. Proteins are macromolecules with many functions, and 20% of our body's mass is. So a big amount of our structure is composed of proteins. And their monomer is amino acids. Just to summarize, the monomer of carbohydrate is glucose or hexose sugar. Monomer of lipid will be fatty acids. And monomer of proteins are amino acids. There are 20 types of amino acids, 20 different types we find. And every amino acid will have a central carbon attached to amino group. This is the group NH2, amino. And there's a carboxylic acid group and unique R group. Earlier, you know that you found this carboxylic acid group or R group in lipid, but I mean, especially that lipid part, we talked about it, but this is something special here, NH2 on your left side, the amino group. But the R group determines the amino acid's property. This is the part that will be responsible for the property of those 20 different types of amino acids. Examples of R groups are hydrogen in glycine, adding the structure in tryptophan, or sulfhydryl in cysteine. Look at that. These are 3 amino acids on your left side. This is the hydrogen responsible for the property of glycine. Then this amino group, amino acid here, and then that's for the tryptophan and polar cysteine amino acid group here, also different from the other amino acids. Amino acids are the monomers, and when they are linked together by dehydration synthesis, and they form the special covalent bond, and those are peptide bonds. Take a look at this. Alanine on your left side, lower part, you see alanine, that's an amino acid. And when it is combining with glycine, alanine plus glycine, one molecule of water is removed and they are synthesizing a new molecule dipeptide here. So this is dehydration synthesis. 2 of those amino acids will make dipeptide here on this side. On the other way, opposite direction, it will be the hydrolysis. Dehydration reaction results in formation of peptide. They're joined by peptide bond. That's the bond you see in between at the middle. This peptide is composed of 2 amino acids and called dipeptide, held together by 1 peptide bond. What about polypeptide? Poly means many. When 10 or more amino acids are joined together, they will form polypeptides. And proteins consist of 1 or more polypeptide chains folded into distinct structure. That is the part coming up soon. But let's see what are the examples of protein structures. Fibrous proteins are long strands found in hair, nail, tendons, and bones. These are fibrous. Globular proteins are spherical, functions as enzyme, hormones, cell messenger. So a lot of those important functions are here. And in future, we are going to discuss their functions in detail. But now, for now, let's take a look at this level of protein structure. With amino acids, when they combine together by dehydration synthesis, proteins are formed. But their level are different: primary, secondary, tertiary, quaternary. Primary structure looks like this. You see every bit here on this string is the amino acid, and they are bound together by peptide bond. And these amino acid sequence of the polypeptide held together by that covalent peptide bond, it's a very simple. This is called primary structure. So multiple amino acids, they are making bonds, which is the peptide bonds, and this is primary structure. Now, in secondary structure, these polypeptides are going to fold over one another, like this. You see, they are folding, they are making a helix-like structure on your right side, and on the left side, they are making a beta pleated sheet. So what is happening here? This, at the primary structure, they only just make a long chain of amino acids. But now they are either making a right-handed spring alpha helices or making a beta pleated sheet like Venetian blinds, which looks like the left one. Right? This is the beta pleated sheet, Venetian folded structure, or as I said, the alpha helices, which is like a strings of spring-like structures, right-handed spring. So these are the secondary structures. We are not done yet. Let's go to tertiary, level 3. This structure refers to three-dimensional shape of polypeptide chain, contains twists, folds, and coils that form the alpha helices and beta pleated sheets, held together by electrostatic ionic or hydrogen bond. So now, from two-dimensional, they are becoming three-dimensional structure. You see here, this is the those they are making these proteins. They are making a globular protein on the left side that works as an enzyme, or fibrous proteins found in hair, nail, those structures. But the point is, they are making a three-dimensional shape. These are tertiary structure. Now, the last level, level 4, this time quaternary. You know the word quarto? Quarto means 4. So primary, secondary, tertiary, and quaternary structure, level 4, when these polypeptides are going to, there will be more than one polypeptide chains you see here, and they have their own primary, secondary, tertiary structure, but they will combine together. The assembly of two or more polypeptide chains into the functional proteins, and this is the quaternary structure, right? So if we summarize this part, this is what happens. At the top part of this slide, you see primary structure when the amino acids are just bound in a string like this, making a polypeptide chain. In step 2, they become three-dimensional. These are making the beta-plated sheet or alpha helix-like pattern. Step 3, tertiary structure, they make these three-dimensional structures here. And then number 4, more than one of those primary, secondary, and tertiary structures, 2 or more of them will be needed to make this quaternary structure. All right, so last point to note is the protein's denaturation. Denature, they lose their natural structure. They lose their shape due to heat, pH, or chemicals. Just think about the white part of the egg. You have taken a fresh egg, and the white part is the protein. When this part we apply heat or some pH is changed or chemicals are applied, that will change. That's how it works with any kind of proteins like fish, meat, egg white, and this denaturation will happen. At that point, the protein function is lost, and the enzymes lose their ability to bind substrates and carry out function when there is protein denaturation. That is all for lesson 7.